In this video, we are going to demonstrate about the enzyme immobilization kinetics. Enzyme immobilization is nothing but arresting the movement of an enzyme in the solid platform. So there are different methods that we can use to immobilize the enzymes. For example, there are two major categories. One is surface bound enzyme immobilization, another one is entrapment based enzyme immobilization. Whereas the surface bound immobilization, the enzymes are bound on the surface of the solid platform through either covalently interaction or non-covalent interaction. Whereas the non-covalent interaction is highly leaky, so it is not highly suitable for surface immobilization. Only the covalent interaction is highly suitable for surface immobilization. But it is pretty costly in comparison to the other method. The other method is the entrapment method. So in entrapment method, there are two types of entrapment method. One is a gel entrapment method, whereas the enzymes are bound inside the enzyme matrix. The matrix is prepared by the various type of matrix, either based on pH or it may be a calcium alginate. So where the enzymes are trapped inside, there is no chemical interaction between the solid platform and the enzyme. Only physically is entrapped inside your matrix. Whereas another one is called a membrane encapsulation. So here the enzymes are sandwiched between two membranes. So the advantage of using membrane entrapment method or encapsulation method, so the membrane has a semi-permeable property, so it is allow only substrate to go into the access the enzymes, whereas other particles are filtered out. So that is the advantage of using membrane entrapment method. Each of these method has its own advantage and disadvantage. Whereas the surface immobilization is pretty costly, but it is covalent based surface immobilization is not leaky, so it is used for a many particular time. And also surface immobilization has an advantage, it is not having the mass transfer limitation, so the substrate can easily access to the enzyme which is present at the surface. Whereas in comparison to the other two methods, the substrate has to penetrate into the matrix in order to access the enzyme. So that is causes a severe limitation for surface substrate movement into the enzyme matrix in order to access that. So that is a major disadvantage of using a matrix bound. On the other hand, surface immobilization is pretty costly, whereas the encapsulation method or entrapment method is easy to perform in the lab. So why we need to do the enzyme immobilization? What are the advantages? There are three main advantages for enzyme immobilization. The first advantage is we can reuse the enzyme as much time as we want. So enzymes are not consumed during the reactions. In the reaction, after the product is formed, enzyme can be present in the same form, so we can utilize it back. Whenever we use a free enzyme, separation of enzyme from that reaction product is a major task. So it is not easy to do, it is very costly procedure is involved. Whereas if you are immobilizing an enzyme in a bead or in maybe a solid platform, by through filtration you can just remove the beads so that we can reuse that enzyme. So that is a major advantage of using enzyme immobilization. So the second advantage is it is always increase the stability of the enzyme. So whenever we do a matrix entrapment method, so the same enzyme can be used in high temperature or high pH. So that is another advantage. The third advantage, since the enzymes are attached to a membrane or attached to the matrix, so it is mimicking the membrane bound conditions. So whenever we isolated or work with the membrane bound enzymes, by mimicking this, so we can able to study or investigate. So it always is a technically a problem whenever we isolate a membrane bound enzyme, and if you are working with a free form, it unable to mimic or unable to resemble the original in vivo condition. So in order to reinstate that, we are using enzyme immobilization as a tool in order to mimic the enzyme membrane bound conditions. So these are the three advantages why we are using enzyme immobilization. So what are the disadvantages? The main disadvantages in enzyme immobilization is the activity of an enzyme. So as I already mentioned, whenever we entrap the enzyme inside the matrix, the substrate has to penetrate through this matrix in order to access the enzymes. So this is caused the severe mass transfer limitations. So not all the substrate is able to penetrate. So in comparison to the free enzyme, whenever we do enzyme immobilization, so there is a possibility that the enzyme activity comes down. So that is a major crucial point here. So how we can access the enzyme activity? The enzyme activity can be measured through the rate of reaction. What is rate of reaction? Rate of reaction is number of product produced per unit time. Say if I am having 10-minute reaction, within that 10 minutes, how many product are producing? So that is nothing but the rate of reaction. So simply we can call a dP by dt. To evaluate the enzyme immobilization kinetics, we need to perform an experiment. The objective of the experiment, we are going to compare the enzyme kinetics between the free and immobilized enzyme. So enzyme kinetic means we are going to check the rate of reaction. Later we are going to estimate the Michaelis-Menten parameters. So the material that we are going to use for this experiment is, the substrate that we are going to use is a starch, and the enzyme that we are going to use is alpha amylase. So the alpha amylase enzyme convert the starch into glucose. So glucose is a product here. So to calculate the rate of reaction, so we need to estimate the glucose concentration. So we are using DNS method to estimate the glucose concentration. So once we calculate the rate of reaction, we are going to compare the rate of reaction between free and immobilized enzymes. So two experiments that we need to do. So one, at different substrate concentration, we are going to calculate the rate of reaction with the help of a free enzyme. Experiment number 2, we are going to repeat the same, but instead of free enzyme, we are going to use the immobilized enzyme. So at the end of the experiment, we are going to get two set of data. One is for the free enzyme, another one is for the immobilized enzyme. So once we get the data, we need to calculate the rate of reaction. Once we calculate the rate of reaction, there are two graphs need to be plotted. One is called plot. Another one is called LB plot. The plot is a non-linear graph, whereas the LB is a linear graph. So first we will see the plot. Plot drawn between V versus S. V is the rate of reaction, which is in y-axis, whereas S is the substrate concentration or the residual substrate concentration in x-axis. So if I draw the graph for the free enzyme, so we come across like this. So the maximum rate of reaction that we achieved, we call that is a VM. So VM is nothing but maximum rate of reaction. So this is one of the parameter, whereas the second parameter is called Km. Km can be calculated like is shown in the picture. So first we need to calculate Vm, then we need to calculate Vm by 2. So whatever that Vm by 2 at that point, what is the substrate concentration is nothing but is equivalent to Km. So once we drawn this graph for a free enzyme, over this graph you need to plot the immobilized enzyme. So the immobilized enzyme is shown in the red line. So what we are expecting is the immobilized enzyme either it equivalent to the free enzyme graph line, or it might be less than what we have achieved for the free enzyme, like you see in the picture. So this is the difference in the Vm is what we need to calculate. So similarly the Km also might vary for immobilized and free enzyme. So though we get the data that we need, both the parameters, but it is comparatively less accurate than the linear graph. So we need to draw the linear line, that is LB plot, like you see here. So the red line is shown here is nothing but for the immobilized enzyme, whereas the black line is for the free enzyme. So now you can see there is a change in the order. Why? If you look at the y and x axis is nothing but 1 by v, and x axis is nothing but your 1 by s, and the y-intercept of this graph is equivalent to 1 by vm, and the slope of these lines is equivalent to the km by vm. So from this you can calculate the vm and km of the both the line. So this is how you need to calculate the km and vm for both free and immobilized enzyme once the experimental data that you got. So now we see what is the experiment, how to perform the experiment. Once we get the experiment is done, you need to interpret the result with the 2 points. The first one is that activity is decreased because of the immobilization. It is very important when you are doing enzyme immobilization, because enzyme immobilization might adversely affect the activity, but what percentage that affects? So that is the question here, whether it is decrease, and if it is decrease, what is the ratio or what is the percentage of decrease in the activity. That is your interpretation of this experimental data point, experimental setup, right? So now we'll see the how the experiment is done.